All right, we can go ahead and start our webinar. Looks like everybody joined. Good afternoon, everyone, and good morning to whoever is joining from the other side. Today, I'm going to be the host for this webinar. I am Monica Johnson from MCE, and today we have a very wonderful speaker who is going to talk to us about TGF-β signaling and organoid fibrosis. And she is Dr. Joyce Thomas Gavali from Thomas Jefferson University in Philadelphia. Moving forward, I will give a little bit introduction about her, and I will also share the webinar agenda first, and then we'll move on from there. Welcome everyone for the webinar, and we're so excited for this webinar series. As I mentioned that we have a very wonderful speaker today from Thomas Jefferson University, and I would like to introduce her, and she's going to give the main presentation, and after the main presentation, we will head on to the Q&A session where you can drop in your questions in the Q&A box. And then after that, we will have some surprise announcement. And then after the surprise announcement, we will do some promotional events and we'll end our webinar from there. Well, let's go ahead and let me introduce our wonderful speaker today. Today our speaker is Joyce Thomas Gavali. She is from Thomas Jefferson University in Philadelphia. She serves as a president of Postdoctoral Association, where she organizes events to engage the team members better and make them feel more welcome in the lab. She is currently working under Dr. Jiasen Sun as postdoctoral fellow in cardiovascular diseases. She integrates her expertise in molecular mechanism of excitation, contraction, in coupling in cardiac fibrosis and pulmonary artery hypertension to extend the boundaries of science. Before joining Thomas Jefferson University, she worked as a postdoctoral scientist at Smith Heart Institute, Cedars-Sinai Medical Center in Los Angeles under Dr. Goldhaber. She broadened her origins by experimenting with the heart failure model to understand the role of the PMCA1 gene in excitation, contraction, and coupling. Hailing from a small town in India, she didn't take an easy way to reach her goals. Always daring and bold, she followed her passion for earning a doctorate in pharmacology at San Vasto in Mexico under the renowned Dr. Jorge Sanchez. Her graduate studies focus on regulation by reacting oxygen species of store-operated calcium release in adult rat myocarditis preconditioned with dioxide. She also has she's also a member of the American Heart Association, and she wears many hats in her role and everyday life. Now I will turn our time to our wonderful speaker, Dr. Gavali.
Thank you so much, Moni. Thank you so much for the beautiful introduction. I think, let me see. Let me share my screen. Yeah, can you see my screen?
Yes.
Yeah, sounds good. So hi, everyone. I would like to thank Monica and Medcam Express team for giving me an opportunity to come and share my science with you all. So moving forward, let's start with the topic I have chosen to talk on today. So it's TGF-β signaling and organ fibrosis. So before we move on, let's see what fibrosis is. So fibrosis it's actually one of the leading cause of cardiac death across the globe. So fibrosis is a condition that develops slowly, but eventually leads to tissue degeneration, which has like very devastating effects on various organs, such as heart, lung, liver, kidney, and also skin disorders. So fibrosis happens when there is an excessive deposition of extracellular matrix or extracellular matrix components such as collagen 1 and fibronectin. Like these are two main proteins which actually is very important to keep the extracellular matrix uh, like in a good shape. However, when there is like excessive deposition of ECM, they release this protein. And so preserving tissue integrity and architecture in response to any injury is very important and evolutionary advantageous mechanism. However, when there is like too much reparative process or when there is like continuous reparation of this system, so it does not only restore the necessary connective tissues, but it also remodels the site of injury with an like excess of extracellular matrix deposition, with the impairment of organ function, and this process is commonly called as fibrosis. Let's talk about like the common manifestations and the etiologies of fibrosis. As I said earlier, there are like different organs which is being affected by fibrosis. So I would like to discuss some of them here. Let's start with the lung. So there are like so many different reasons why the lung why there is a fibrosis in the lung and like different organs. So in lung, fibrosis can happen because of idiopathic pulmonary fibrosis, chronic infection, hypersensitivity, and radiation. In liver, the fibrosis can happen because of non-alcoholic fatty liver diseases, chronic viral hepatitis, alcoholic liver diseases, and cholestatic liver diseases. In intestine, it can happen because of the secondary inflammation, bowel diseases, and radiations. Let's come to the heart, like the main important part I am talking today about, and it's like my expertise. In the heart, cardiac fibrosis happens because of so many different insults, such as ischemia, myocardial infarction, cardiomyopathy, hypertension, and aortic stenosis. In kidney, it happens because of hypertension, diabetes, atherosclerosis, recurrent infection, and glomerular diseases. However, with the skin, it happens because of autoimmunity, because of some drugs, traumatic, and hypersensitivity. Then let's dig in about like how like what is extracellular matrix and what kind of extracellular matrix overproduction causes this fibrosis. So I don't know if you can see the pictures really very well. I have like zoomed out some areas or some fibrotic areas of some of the organs. As you see in liver, kidney, heart, and the lung, you see there is like change in the architecture of these organs. So it happens because of excessive production of cytokines. There are so many different types of cytokines, such as pro-inflammatory cytokines, Th1, Th2. Some of them are like interleukin, TGF-β, TNF-alpha. There are also some chemokines such as CCL families. There are like so many different isoforms. I don't want to like tell one by one all the isoforms here. And also some growth factors such as TGF-β, and some extracellular matrix proteins such as collagen, fibronectin, CTGF, some others. So excessive production of extracellular matrix is an hallmark for fibrosis, which then causes loss of normal organ architecture and function. So let's talk about the mechanism of fibrosis. So fibrosis causes the main cells which are like or the master regulator of fibrosis are the myofibroblast. However, so there are like, you know, right, cells are our body has like so many different cell lines, so many different cells. So let's talk here like one by one. There are like so many different cells such as vascular smooth muscle cells, mesothelial cells, fibrocytes, resident fibroblast, epithelial cells, endothelial cells, like all these cells with the specialized cells, they undergo a phenotypic switch through different pathways. Let's take an example of fibrocytes. Because of some insults or some stimulations, fibrocytes undergo infiltration and activation, and it then turns into a myofibroblast. Same like this, all different cells, either through proliferation or the transition from one cell to another cell, such as EMT, endothelial to mesenchymal cells transition. All these different transitions causes these cells to switch to myofibroblasts, and these myofibroblasts are the main cause of fibrosis. These are the main cells which are mainly involved in causing fibrosis. Um, let's talk. I would just like to dig deep, not super deep, about how epithelium causes this phenotypic switch from normal cells to the myofibroblast. So epithelium is the first hit whenever there is an injury. So epithelium, injured epithelium, through three different pathways. One is TGF-beta signaling, which is like the main pathway I'll be talking about. Another one is inflammatory signaling. I don't know much about the inflammatory signaling because that's not like my expertise. I just brought up this to have a clear idea that not only, sorry.
Go ahead.
Yeah. So not only TGF-beta signaling is involved in fibrosis, simultaneously there is like some other signaling as well, inflammatory signaling and Wnt signaling. I don't want to talk a lot about the other signaling, but there are like some cross-talks between TGF-beta and other signalings during the fibrosis process. I will explain in the coming slides how these different crosstalks can affect the fibrosis. So TGF-beta is the master regulator of fibrosis. The end product of fibrosis is a lot of TGF-beta being synthesized. So let's talk about the overview of TGF-beta signaling. So generally TGF-beta signaling, there are two types of TGF-beta signaling. One is canonical pathway and another one is non-canonical pathway. I'll talk about the non-canonical pathway in next slide, but however, I would like to talk a little bit more about this TGF-beta. So TGF-beta, like in normal condition, is always associated with let me turn the laser pointer. Okay, it is always associated with this LTBP. I think it's like latex binding protein, and also with like LAP. Because of some stimulation or some insults, these association breaks down, causing TGF-beta go and bind with the TGF-beta receptor. So once TGF-beta is activated, it further activates TGF-beta receptor 1 and 2. These are most common isoforms in human. However, whenever these two receptors are activated, it further phosphorylates SMAD2 and 3 in the cytoplasm. And once this is phosphorylated, they translocate from cytosol to the nucleus by further activating some fibrotic genes such as alpha SMA, FN1, like fibronectin and collagen. However, there is one more SMA family here, which is SMA7, which can actually inhibit this interaction. If you are using any activator, or if you overexpress SMAD7, you can actually inhibit this phosphorylation of SMAD and inhibit the translocation of these proteins to the nucleus by inhibiting fibrosis. I'll talk about it in the coming slide. So that's all about the TGF-beta. in fibrosis, and as I said before, that there is always a crosstalk between TGF-beta SMAD and some other pathways in the tissue fibrosis. The pathways which I am focusing in my studies are basically the ERK pathway. I don't really focus on the Wnt signaling or the Notch or ALK1, but yeah, there are indeed all these other pathways which can also be regulated through TGF-beta 1. As you see here, like TGF-beta also activates RhoA1, GRB2, ERK, which then further phosphorylates this SMAD and then activates the fibrotic gene pathway, or it shuts on the fibrotic signaling. Uh, um, Before I start showing my science, I would just like to show you this, that there are some like potential therapeutic strategies which is like being used to inhibit the TGF-beta SMAD induced tissue fibrosis. I have shown here in this figure with the red arrows, the inhibitory red arrows. For example, there are some neutralizing antibody which can actually neutralize TGF-beta 1. And also there are like some decorin and some other receptors which can actually block this out. If you block this out, it will not further activate the receptors and then smart activation. There are like some more. You can actually also activate some add some receptor kinase inhibitors to block out the smart 2 and 3, or the smart 7. If you overexpress smart 7, it will also block the phosphorylation of smart 2 and 3. Apart from smart 7, there are also some other inhibitors which can actually being used to prevent the fibrosis. Um, so today I will be, let's move on to the project I have been working to prevent the fibrosis. So my project or my science basically is on understanding role of LMCD1 in organ fibrosis. I I am mainly focusing on only heart and lung as of now. In future, we may think of moving to a different organ, but as of now, we have only done science with the heart and the lung. So let's talk about LMCD1, which is also known as limb and cysteine rich domain 1. So LMCD1 is a member of a limb family protein, and because it is a limb family protein, this limb family encodes, this family has like a zinc finger proteins, which is why this protein is mainly involved in protein-protein interaction. And this protein may act as a co-regulator of transcription along with other transcription factors. And it also plays an essential role in cell signaling and regulation of gene expression. So previous reports have already published some data showing how LMCD1 is associated with so many different diseases. Some of them are like cardiac hypertrophy. LMCD1 is involved in the regulation of cardiac hypertrophy. Also kidney diseases, skeletal muscle diseases, interstitial lung diseases, hepatocellular carcinoma, and mitral wall prolapse. Um, this is one of the data I got from Brian et al. They published this data in 2010, where they challenged the mice without HIG binding to induce the hypertrophy. And they just take the expression of LMCD1 at protein level and mRNA level at different time points. As you see here, after 1 week of post-aortic binding, there is an increase in LMCD1 protein, also in the increase in the transcripts of LMCD1, which is actually very interesting. Moving on, they also did some experiments by overexpressing LMCD1 in the mice. As you see here, these are like some sham animals and without it binding animals. So these are like the pictures of some heart samples. So in wild type mice, when you in the sham, when you expose them to certain stimulus, there is no change. However, you can see there is significant hypertrophy. The heart is bigger in size compared with the wild type one. However, when you overexpress LMCD1, there is more hypertrophy. The heart is way bigger. So, and also there is like some histological staining showing that there is indeed a change in the histology of these heart sections when they overexpress LMCD1. So by keeping these on my mind, we plan to check the LMCD1 in cardiac fibrosis and remodeling. So let's talk a little bit about what is cardiac fibrosis. Cardiac fibrosis is a common feature of many cardiac insults, such as myocardial infarction, hypertension, diabetes, or hypertrophic cardiomyopathy. It is also marked by an expansion of cardiac interstitium due to excessive deposition of ECM. Till now, there is no anti-fibrotic therapy exists, so which is why we are aiming to elucidate the molecular mechanism and develop the novel treatment strategies to reduce the fibrosis. Here I have shown a simplest figure showing how does the cardiac fibrosis happens. This is the heart, and because of some stimulus, cytokines, or growth factors, the fibroblast in the cardiac fibrosis are turned into myofibroblast, causing excessive deposition of extracellular matrix and then leading to the fibrosis. So now
Joyce?
let's talk about, yeah.
There are a few people joined again, so joined. I would like to just welcome them here and just
Yeah,
let them know.
sure.
We are now listening to Dr. Joyce give us her presentation here. And welcome. You can go ahead.
Yeah, thank you. So now I'll be talking about the role of LMCD1 in TGF-beta signaling and cardiac fibrosis. So we have done like very simple experiments initially just to see if LMCD1 does regulate in presence of TGF-beta. So we have incubated human cardiac fibroblast with TGF-beta. You can see we have incubated with TGF-beta at different dose, different time points, because we wanted to see or pick up the good time point to further study. Um, so you can see after 3 hours of TGF-beta, there is an increase in LMCD1, and we also wanted to check whether there is an increase in fibronectin or whether there is an initiation of fibrosis or not. So we also did check some fibrotic markers such as fibronectin and collagen 1, and we see there is a significant increase of LMCD1 and some other gene in response to TGF-beta. And then I also have done some immunofluorescence to see how the cells behaves in response to TGF-beta. As you see here, these are like control and TGF-beta. So there is an increase in LMCD1 and collagen. So I just like stained the cells, human cardiac fibroblasts with anti-LMCD1 and anti-collagen 1. And then I just did confocal microscopy. And you see there is an increase in collagen 1 and LMCD1 in the cells treated with TGF-beta. I also have checked fibronectin, and there is an increase in fibronectin as well. Moving on was like, okay, we know there is an increase in LMCD1 and also there is an increase in fibrotic gene. Next, we wanted to check if we knocked out LMCD1, does it attenuate fibrosis? So we had used lentiviral bearing SH-LMCD1 to knock down LMCD1, and we have chosen 24-hour time point because that was the best time to see the fibrosis and also change in LMCD1. As you see here, there is an increase in LMCD1 in control in response to TGF-beta. However, when we knocked out LMCD1, this was completely the effect of or the expression of FN1 or collagen was completely gone, which means if you knock out LMCD1, we saw that there is a decrease in fibrosis. We also did some qPCR experiments to check out different genes to see if the Western blotting data is consistent. So we have seen an increase in collagen 1A1, collagen 3A1, fibronectin, SMA, and CTGF. We see there is an increase in the mRNA expression of all these genes. However, when we knocked out LMCD1, the effect of TGF-beta was completely blocked. Next, we wanted to check in vivo if our hypothesis is true when we do the experiments in the mice, because previous was like just the in vitro some cell line. So we have challenged these mice with angiotensin. So we have just like did osmotic pump implantation on these mice with angiotensin, and then after 4 weeks post angiotensin, we sacrifice the mice. But before we sacrifice the mice, we have done echocardiography to check actually the cardiac function. And as you see, there is actually increase in left ventricular anterior wall thickness, systolic and diastolic, and left ventricular posterior wall thickness, systolic and diastolic, in control mice in response to angiotensin. However, when if you see in knockout mice, there is a decrease in the wall thickness, which means LMCD1 deficiency does affect the cardiac remodeling. It brings down or it prevents the cardiac remodeling. I also have done some Western blotting to check the expression of LMCD1 and some other fibrotic genes. And I got the consistent data with the in vitro data that there is an increase in LMCD1 in wild type mice in response to angiotensin. However, the expression of collagen fibronectin were like inhibited from the knockout mice. So then as I showed in the previous data about the signaling pathway, the canonical one, the phosphosmad one. So we just like wanted to check what happens to this signaling pathway if you knock out LMCD1. So as you see here, there is an increase in the phosphorylation of phosphosmad in early treatment of TGF-beta 1. However, this was completely gone when you knock out LMCD1. And then I'll move on to the pulmonary fibrosis. So let's see what pulmonary fibrosis is. So pulmonary fibrosis is a lung disease that occur when lung tissue become damaged and scarred. It is an interstitial lung diseases. I will talk to you about what interstitial lung diseases in the coming slide. So interstitial lung diseases are a group of conditions that cause inflammation and scarring around the tiny air sacs in the lung. Normally, the thin, lacy walls of air sacs in the lung are no longer thin and lacy, but it gets thick, stiff, and scared, which is also called fibrosis. So with this scarring, the lungs architecture becomes stiffer and less efficient at delivering oxygen into the bloodstream. In addition, stiffness or fibrosis of the tissue makes it more difficult to expand the lungs, and breathing requires more effort. And this additional demand of energy or work leads to shortness of the breath. So interstitial lung diseases is an umbrella term for all different kinds of diseases related to the lung. So I don't want to talk like much detail in about it, but yeah, fibrosis also comes under these interstitial lung diseases because, as I said, it's an umbrella term. Let's talk about the TGF-beta signaling in lung fibrosis. So TGF-beta activates, so this mechanism actually is pretty much same with the cardiac stuff. So TGF-beta signaling activates SMART pathway by further activating fibrotic genes such as SMA, collagen 1, and then it causes the phenotypic switch from fibroblast to myofibroblast, and then causing the excess deposition of ECM and then finally leads to the lung fibrosis. So, and then let's talk about the role of LMCD1 in pulmonary fibrosis. So here we treated human lung fibroblasts with TGF-beta, and we have seen an increase in LMCD1 and also increase in collagen 1 and fibronectin. We have also done some immunofluorescence study to see And we found that LMCD1 is increasing in response to TGF-beta, and also there is an increase in the collagen 1 in response to TGF-beta. However, when we knock out LMCD1 from human lung fibroblast, so when we knock out these genes, especially LMCD1 from fibroblast, we saw that the effect of TGF-beta was blocked. So to check or to study the in vivo findings, we have challenged these mice with bleomycin. So we have given the mice bleomycin and we sacrifice the mice after 14 days. As you can see here, there is an increase in LMCD1 in response to bleomycin. Also an increase in the expression of collagen 1 and fibronectin 1. However, when we knock out LMCD1, we see that the collagen expression was coming down and also the fibronectin. We also have analyzed this data. As you can see here, there is significant decrease in the fibrotic genes when we knock out LMCD1 in response to bleomycin. We also have checked some transcripts or the transcriptional regulation of some genes like LMCD1, fibronectin, collagen 1, and collagen 3. They all are going down when you knock out LMCD1. We also have done some histological stainings. So I did use 2 different staining. The first one is Yachi staining and the another one is the MASH and trichrome. As you see in Yachi staining, in wild type mice, in response to bleomycin, the lung is more patchy. You can see all those patches here. However, in knockout mice and in knockout in response to bleomycin, this patchiness was like almost gone. And the second staining we have done is the MASH and trichrome, which actually stains collagen in blue. As you see, there is like blue staining. This is all collagen, which is stained by the MASH and trichrome. So there is more collagen in wild type mice in response to bleomycin. However, when you knock out LMCD1, the collagen 1 is less. We also did check the downstream signaling pathway, same like cardiac fibrosis. There is an increase in phospho-SMAD2 in early response to TGF-beta, which is completely blocked when you take off or when you knock out LMCD1. We also did check some pathways just to double confirm if by any chance some other non-canonical pathways also involved. However, we did not see any change in the phosphor and the total ERG. So overall, like talking about the lung summary, what we have found so far was in TGF-beta signaling, there is an increase in SMART2 and 3. However, after in LMCD1 mice, since there is no LMCD1, this pathway is being blocked and it is further blocking the excess of extracellular matrix deposition by inhibiting some of these fibrotic genes. So in summary, in fibrotic stimuli, there is an increase in LMCD1, and this increase in LMCD1 is overactivating the SMAD2 to 3, causing this SMAD2 to 3 going to the nucleus and activating these pro-fibrotic genes, such as collagen 1, 3, fibronectin, and CTGF. However, when you block LMCD1, there is no longer LMCD1 to activate this pathway, or this LMCD1 is actually blocking SMAD when it's not in the body, and by further blocking the extracellular matrix production and activation of these pro-fibrotic genes. So I would like to thanks my team for always helping me support. Would also like to thank Thomas Jefferson University for supporting the science. So this is my team. This is my PI, Dr. San. This is me, my other lab mates, Dr. Noprat and Dr. Shujan. That's it. I'm open to take questions.
Thank you, Dr. Joyce. We appreciate your time and wonderful presentation. Now we will head on to the QA, and you can drop in your questions in the QA. Okay, we have a question by Dr. Lee. How does LMCDI1 activate SMAD2 or 3?
SMA is small, right?
Yes.
Okay, that's actually a very good question. We actually don't know yet how LMCD1 is activating this. SMA is actually a very important marker to see the phenotypic switch from fibroblast to myofibroblast, right? We have seen SMA going up and it's been going down when you knock out LMCD1, but we don't have um, result yet showing how LMCD1 does activate SMA. We don't have the data yet.
Okay. Somebody else, Bide, asked a question. Curious to learn more about how the speaker identified the LMCD1 targets. How can we rule out effects on LMCD1 knockdown on TGF ligand or TGF activation or etc.?
That's actually a very nice question. Since TGF-beta is like a big complex, we have only studied smart signaling. So I would say LMCD1 is blocking out these smart and 2 to 3 pathways. I have done some experiments to check, not directly, the TGF-beta. We did not see any change in the expression of these proteins when you knock out LMCD1. So I would say we don't see a direct effect of LMCD1 knockout and TGF-beta, but yeah, there was only change in the smart signaling.
Okay, so there's another question by John Chen. He's asking, what is the relationship between LMCD1 expression levels and the severity or progenesis of disease such as heart failure or specific cancers?
Okay, I am not sure about the cancer because cancer is not my expertise. Yeah, but heart failure is late stage diseases. There is one article showing LMCD1 expression decreases in the human patients with the heart failure. So I would say since it is like late stage disease, maybe LMCD1 compensates by decreasing its expression. That's the only answer I could give you right now, but we are not studying the heart failure as of now because it is late stage diseases. So we only right now focusing on the fibrotic phase. But yeah, we don't see any change of LMCD1 overexpression or giving more LMCD1 in the heart failure things.
Okay. Another question by Meenakshi. How are fibroblasts cultured? Is it primary culture?
Yes. Yeah, we do use basic fibroblast medium. So we bought these cells from the company. We did not isolate in the lab. They are human cells, so no. We bought these cells, the primary cell line from the company, and I did use the company suggested a basic culture medium.
Good. I'll go ahead with like 2 more questions and then we can move further because there's so many questions and I have to pick and choose. Okay, somebody's asking, is this data published? Could the speaker provide the reference?
No yet.
Okay. Okay, last question, or second last, maybe we can go one more. I see you worked in human cardiofibroblast cells. Which company did you use your promo cell or cell applicants? Also, till which passage the cell responded well as they are primary cells?
Yeah, so we bought cells from Lonza. We also bought from PromoCell once. I would not use cells beyond P5 because they don't really keep well if I use them beyond P5. Unlike human lung fibroblast, I can go beyond P5, but cardiac fibroblasts are very sensitive.
Okay. Okay, the last question. We can end the QA with this. Thank you so much. Could you discuss the potential therapeutic strategies targeting TGF-β1 for managing um, fibrotic diseases and what challenges might be faced in developing these treatments?
Yeah, that's actually a wonderful question. Yeah, TGF-beta, you can actually target TGF-beta to reduce the fibrosis, but if you target TGF-beta, it will increase the risk of autoimmune diseases, which is why these days, instead of focusing on TGF-beta, they are focusing mainly on the downstream signaling pathways. Like I am doing, I'm basically focusing on phospho-SMAD 2 and 3.
Okay, that sounds good. I think we can end our QA here. We really appreciate everybody's participation, asking questions to Dr. Gawali here. We will move ahead and in our sessions, and we want to thank Dr. Gawali for a wonderful webinar today. So here we're at the surprise giveaway, and whoever joined, please register in our website to get 10% coupon. You can just scan this QR code here and get into our website as a new account, as a new user. And whoever already has an account with us, we will go ahead and give you another 10% coupon for registering with us. And now to the next part, we're gonna go and tell the eventual promotions from our marketing team here. First, let's start with the left-hand corner on the top. Let's focus on the left-hand corner. Regarding our compound screening libraries, you can see on the top, we can be able to customize your libraries according to your specific needs. The price per compound determined by the quantity you select, with the prices as low as 2.2 per compound. Additionally, if you purchase the 8-channel handheld screw decapper, you can enjoy a 30% discount. So if you're planning to restock, now is the best time. Moving on to the top right corner for the recombinant protein-free samples. We have a variety of applications, including cell growth and differentiation, cell signaling, studies involving disease onset and progression, biopharmaceutical target discovery and protein structure, and as well as function analysis. Now you can apply up to 5 free samples. Now moving on to the left bottom corner. In addition to free samples, purchasing any of our recombinant protein or our best-selling kits, like if you are working like Dr. Gawali with researching fibrosis mechanism or developing anti-fibrosis therapies, don't miss out on our basement membrane matrix, which is now available for 30% off discount. Finally, moving on to the bottom right corner, where you can see the citation award for $500 voucher. If you've successfully published a paper using our products, you could be eligible to apply for our $500 reward voucher. And many more gifts. On the bottom below that, you can see if you register and collect points, when you redeem it, you could be able to get different gifts like $5 coupon and 10% off coupon and mouse pads and even Amazon gift card and Starbucks gift card as well. So I would like you to go ahead and register. And if you have any questions, please reach out to me and to our team. We will always be available. And we really appreciate your time today. And we really appreciate Dr. Joyce here for being there today and giving us wonderful insight on TGF-β and fibrosis. And thank you so much. And this is how you can reach out to us. We would be looking forward for future webinars. And this is our email and the phone numbers, how you can reach out to us. And we will be looking forward for the next time you can attend. Thank you so much.